Awesome. Thank you all for coming to this large airplane hangar here about sex chromosomes. So, in the event that you don't find this talk very interesting, you should come up during the break and look at this heliderma skull. So, I brought a prop to accompany it. I'll also preface by saying that we had anticipated to do a bunch of evolutionary analyses when I submitted this title. That's the Evolution Conference chiding me on not having done so much evolutionary analyses. But what I do hope to do is tell you a little bit about why we think the Gila monster, Heliderma suspectum, is so interesting, and why it is a terrible model, unlike the turtles that Nicole Valenzuela is looking at. The next closest relative to the Gila monster is the Mexican beaded lizard, which is 50 million years diverged. The next closest is the Komodo dragon, which is 120 million years diverged. And then after that is the Anolis, about 160 million years diverged. So it is a lone branch. There aren't outgroups to compare to that are pretty recent. So I still think we should fund it and study it, but maybe not the best model for looking at transitions in sex determination. So that said, I wanted to also mention a little bit about crowdfunding this genome. This is an adorable sketch someone sent me of the Gila monster, a bit about its sex determination, why we think evolutionarily it will be really interesting, collecting the DNA, and then what we do have some interesting things. So I did go to the NSF table earlier, and they said, do you know about the NSF? And I said, yes, you've denied me funding multiple times for this project in multiple different ways, comparative ways, single ways. We just got no everywhere we looked, and so what we did is we tried to crowdfund it, and we actually were pretty successful. So we raised about $10,000, which, as you all know, is nearly nothing in the realm of doing science, but it was enough to get us started, and it was enough with these 173 backers. So we also gained a collaboration with 10x Genomics, who helped us get the scaffolded reference genome so that then we could also use some of this money to do both DNA and RNA from a set of males and females that we have access to. Oh, yet another reason why the Gila monster is a terrible model. They're protected in the state of Arizona, so we cannot get gonads, we can't get multiple tissues. In fact, we can only get non-invasive tissues here, which is blood, unless one happens to die and you happen to be near it at the right time, which has happened once, and we have those samples in the freezer for future reference. Would I do crowdfunding again? No, I wouldn't. It was a ton of work, and I loved it. I loved interacting with everyone, but it was a tremendous amount of work for a very small amount of money. On average, crowdfunding funds about $2,500. So if you're a grad student looking to fund the last $2,500 of your research, I highly recommend it. If you are a PI trying to fund a lab, this is not a good investment of your time, in my very professional opinion. Okay. So we did also have a collaboration with Dan Beck, who helped us. He wrote this wonderful book about Gila monsters and beaded lizards, and we were able to give those books away to some people. So the thing is, though, this just let me connect with the crowd of people who are interested in Gila monsters. And then you might say, Melissa, I know you a bit. You study mammals and things with placentas. Why on earth are you studying a Gila monster? And I would say, because I moved to Arizona State University, and my next door neighbor lab studies physiology and Gila monsters. And I went to Dale DiNardo, my next door lab neighbor, and I said, Dale, this is so cool. How is sex determined in the Gila monster? Dale, who studied Gila monsters for 30 plus years, had no idea how sex is determined in the Gila monster. Ergo, our collaboration. So what we do know is that there's been work shown that Gila monsters have ZZZW sex determination. So some really nice fish work has been done on this fluorescence in situ hybridization, not mixing species here. And what that means is just that females can produce an egg with a Z or a W sex chromosome. You'll find there's the most eclectic and amazing art with Gila monsters if you get into this field. This is Gila Monster's wedding day with Roadrunner, who also, by the way, has ZW sex determination, but not the same sex chromosome, so unlikely to form hybrids. I won't quit my day job, but I am having a good time here with you all. So why do we care about Gila monsters? So they are this really unique lineage. They evolved from a tropical ancestor that lived with tons and tons of water, and now they live in the desert. So they have developed some unique adaptations to the desert. For example, they can regulate their water usage using their cloaca, opening and closing with their bladder. They're one of a very small number of venomous lizards, but they don't have venom glands. It's just their saliva that has what we consider to be the venom. They're also not deadly. So, and again, we'll make the distinction venomous, not poisonous. You could eat it, but you wouldn't want to. You just don't want to get bit by one. It will feel like flaming lava and you will swell up. There's nothing to do other than sit and suffer until it passes. You probably won't die. There's only been one reported death, and that was from the late 1800s, and he was also drunk and wandering in the desert. So very unlikely that it was due to the Gila monster. I won't let anyone shame our monster that way. We would eventually like to get at their venom and look at venom transcriptomes, but it's remarkably difficult to get saliva off of a Gila monster. So I've been looking into ways that you can pipe onto this kind of plastic straw and have it collected. Great. So other questions that we care about as evolutionary biologists is that there are still remarkably few genomes for squamate reptiles. And there's large discrepancies between what's available for the molecular phylogenies and the morphological phylogenies. And we're hoping that by adding additional genomes, we'll be able to resolve those and really try to understand. So there were two strange conversions, and this one, I don't know why. That should be orange instead of green, but it's not that big of a deal. It's just that Gila monsters are orange, not green. We know that the Gila monsters have ZW sex determination. We know that Komodo dragon has ZW sex determination. We know that snakes, most snakes, have ZW sex determination, as we're finding out now. And we'd like to know if those are convergent evolution or if they're shared ancestry. So I won't tell you the punchline yet. We'll get there at the end. But I hope that we'll be able to determine that by the end. So this is individual number, actually letter L. He is our reference individual. As you can tell, he's very charismatic. I don't touch him. Dale DiNardo touches him, but I do collect the blood from him and 5 other individuals. This is Dale DiNardo, who is a physiologist and does some really great work looking at osmolality and evolution of adaptation to desert environments. And so we collected DNA and RNA from 3 males and 3 females, including our reference individual here. This is my hand for reference. I don't have particularly small hands, but this is a particularly large lizard. And extracting DNA from these samples. You might not know it, but they like to sit in the bath quite a lot. So they all have these little water tubes, and I'd say more than 50% of the time when we go to look at them, they're just hanging out in their bathtubs or pools, whatever you like. So those are 2 of the other individuals that we looked at. Dale has a colony of 18 of them, and colony is maybe a misnomer because they're all in their individual cages here. But they're all collected from the state of Arizona because we've had so much construction. And one of the things that we found out is that if you take a Gila monster, it has a few kilometer home range around its burrow, which it finds when it's a juvenile. And if you remove it from that home range, it doesn't build a new burrow. They just starve to death. And so you can't rehome them once their home range has been demolished, largely by building a foundation for a house. And so they go to Dale, where they spend the rest of their lives in these spas and are sometimes involved in research. They have to work for their spa life. So we did RNA and DNA from 3 males and 3 females, 75 base pair paired end sequence for the RNA-seq and 150 base pair paired end sequence for the DNA-seq. If you do come to Arizona, they won't meet you at the airport, but you can meet them at Arizona State University. There's some on display. And so largely the sequence has been really good, which was great because one of the things I didn't anticipate is the blood coagulated very quickly for some of these individuals. Largely, they don't drink a lot. They are desert adapted. They have access to it, but we would try to draw the blood and it would coagulate in the syringe as we were drawing it, which did not happen when we worked with iguanas or the Anolis lizard. And then we've been looking at the same thing. So Nicole gave an excellent overview. So we're looking at DNA coverage difference in females relative to males. If it's roughly equal to 1, then we think it's probably an autosomal region. And if it's closer to 50%, so half as much coverage in the females as the males, then we expect that that is consistent with being a Z chromosome, because the females should have 1 Z and males should have 2. And then once we identify those Z-linked scaffolds, then we can go back and look at the RNA to see if there's any evidence of dosage compensation. This is work done by Tim Webster, who was a postdoc and is now a faculty at University of Utah, and Grant Denbrock, who's an undergraduate, who planned to go to medical school, but I think we're slowly turning him to the dark side. So, or maybe away from the dark side, I don't know. So the one thing that also didn't come is our confidence intervals, but we have several of these autosomal scaffolds, and some of them quite large. So this one is 60 megabases from our 10x genomics scaffolded genome. So it's nearly a chromosome size scaffold. And this is the RNA-seq, so the female to male ratio of RNA-seq. And there is quite a spread, but we see for all of the autosomal scaffolds, we have about a female to male ratio of 1. I'm showing you all the data rather than compressing it down into that beautiful little line, but I'll just ask you to focus on the line. And then what we do find is we find 5 scaffolds. I know there's only 4 here. We find 5, but I'll show you the 5th one in a minute. We find 5 scaffolds where the female to male DNA coverage is about 50%. And it turns out this is the RNA. The female to male RNA coverage is also 50%. So it doesn't look like in this Gila monster we're seeing dosage compensation across most of those genes. They're much smaller than that first autosomal one I showed you. The scaffolds for our potential Z chromosomes are much smaller. But largely we're not seeing evidence of dosage compensation, except, oh, sorry, that's all of them together. Oh, okay, we're not seeing evidence of dosage compensation, except, there we go. This one, which is really cool to me because it's a whole scaffolded region. We're pretty confident that we haven't mucked up the scaffold, and we see a part of it showing equal expression between males and females, and then some of it being a little wonky. I don't 100% trust that going back up, but it's a potential pseudo-autosomal region. Yes. And with that, I want to thank the people who worked on this, the Gila monsters, for being so generous in donating their samples to us, and instead of spewing venom, let's study it. Just ignore politics completely. Thank you all very much.
So Scott,
We have time for one.
it wasn't a robot. But it was some kind of flicker, right? Yeah.
Absolutely. For all 5 of those scaffolds, the DNA coverage was 50% across the entire scaffold. So that's why, that's why I think it's not a pseudo-autosomal region, because it's not that. Yeah, and we actually didn't go through, what we could do is a windowed analysis, where we look for, this is across the entire scaffold, but we could look for windows where it's 1-to-1 and then drops down to 50%. Yeah. So the question is, in ZW sex determinations, do we see the equivalent of Klinefelter or Turner syndrome, I guess? So multiple or losses of sex chromosomes. And that's been observed. Not in the Gila monster, because you're right, we don't have that much data, but that does happen in other species. I just think, for the reason you point out, we haven't extensively looked